please can you tell us your name and your institution and what you do?
I am Dr. Isabel Palacios. I work at Queen Mary University of London, which is in London, and I work on how cells and tissues organize during organogenesis so that organs develop the right way. I use Drosophila as a model system, and I also use human stem cells derived neurons and organoids. And some of my work is related to neuronal health, so how to keep neurons healthy and how to try to work on avoiding or curing neurodegeneration. We do fundamental basic biological studies towards those aims that I just mentioned.
Okay, what do you think about the FLY-NEUROCAN workshop so far? What has stood out for you during the workshop? It's just day two, but what has stood out for you?
So this is a fantastic workshop. First, I want to really say what a wonderful center the DRTC is. We've been working with them for a long time now, and with Professor Amos before the center was also installed. And I think it's an incredible and really amazing and inspiring work that they are doing. So that's the first thing to say. Regarding the workshop, I'm really impressed by the quality of the organization. The program they've put together is unbelievable. Taking, input or teaching lessons from many teachers and scientists in the world. And I'm really impressed by the participants so far. They've been extremely outgoing, interested, asking lots of questions, excited about the practical that we've been doing. And yeah, I'm really pleased to be here. It's been too long that I haven't been in Nigeria, so I'm happy to be back. And yeah, amazing, fantastic so far.
Thank you so much. So what key message did you hope the participants to take away from your session?
What do I want the participants to take from my session?
Yeah.
Okay. So from my session, I want them to take, I think, 3 messages. One, you can do a lot with Drosophila to understand some basic biological and genetics of human biomedical conditions, diseases. So the fly is very powerful to do a lot and is very impacting, especially if you want to set up a lab here in Africa and you don't have a lot of money and you don't have a lot of, complicated technological requirement or lab, then you can use the fly. It's relatively easy and it's relatively inexpensive and you can do a lot with it. What I want everyone to know is that regarding this workshop, you can use Drosophila to study some aspects of neurodegeneration. So what we are doing, I haven't done much neurodegeneration in my career. It's only the last couple of years. But what we've done is use the fly to understand some aspects of amyloid toxicity, which is related to Alzheimer's disease. And we've shown that if you increase the number of proteins in the neurons that are known as motor proteins, are a bit like the Okada of the cells, the transport thing around. If you increase the amount of kinesins, this is the name of these motor proteins in neurons that are expressing also a pathological amyloid variant from human information, you can rescue some of the neurodegeneration aspects in using the fly for that. And now we are trying to see whether we can use that information to develop, if you want, a hypothesis of how therapies could develop based on motor protein activity. So that's another message. And the last message, which is related to my charity, Drosophila, is that as Drosophila helped a lot of Spanish scientists, I'm Spanish, during many, many years, during my lifetime, and now you can have hundreds of fly labs all over the world with the Spanish group leaders. I think Drosophila, and that's the work that our charity does, can help set up a lot of labs and a good community of scientists interacting with each other here in Africa. And get in contact with us or contact the RTC to learn how to do that. And we can help you, both Drosophila and the RTC.
Thank you so much. So the last question, what are your expectations for the rest of the program and just for early career scientists coming into Drosophila in research?
Oh, that's a big question. So regarding the workshop, of course, 1 week on neurodegeneration, 1 week on cancer is, of course, a great way to get introduced to how the fly can help in those topics, cancer and neurodegeneration, but it's not enough to understand how to work with the fly. So I think the outcome in the end is for students or participants to realize how much you can do with it. and then carry on interacting with us, with DRTC, with Drosophila, with any mentor that they want to interact with, to carry on setting up and trying to then apply what they've learned here to a project that they are interested in. So for me, that's the main thing. I think that covers both. Now just take in everything you can do, think of what you want to use the FLY for and what can the FLY help you with from your own interest, and then develop it further after this workshop with our help. Thank you.
Thank you so much.
Thank you. FLY-NEUROCAN Workshop 2026.